Hi, everybody. I'm Zen Honeycutt from Moms Across America, and we have a very special interview today with Dr. Michael Antoniou from the King's College of London. Thank you very much, Michael, for being on with us today.
A pleasure.
Awesome. Could you give everybody just a little bit of your background, your credentials, and what you're doing right now?
Yes, certainly. Yes, I'm on the faculty of King's College London in the Department of Human Genetics, and part of my research program for the last 5-6 years has been exploring toxicity of chemical pollutants with a special focus on pesticides, including, of course, glyphosate. So, and we published a number of studies on this. I guess our initial landmark paper was where we showed that even an ultra-low dose of Roundup herbicide administered to animals over a long period of time still resulted in serious illness, in this case, of fatty liver disease.
Yes, and we remember that paper came out in January, about a year or so ago. It was very, very damning, actually, for Monsanto and the makers of glyphosate-based herbicides. And today we're going to talk about a new study that shows that glyphosate, well, let me just read the title of the study. Glyphosate primes mammary cells for tumor genesis by reprogramming the epigenome in a TE-T3 dependent manner. And what that means is that glyphosate can cause cancer with the secondary risk factor that you're going to talk about today. And this is extremely serious. Breast cancer is on the rise all across the country. I believe it's one of the top killers of women in America. I know heart disease is one. And we want to get to the bottom of this because Monsanto continues to claim that it's safe. And we know that it's not. And so can you please explain, what does this new study tell us exactly about glyphosate and breast cancer?
Yes, this study for me is really quite unique so far. What this study is showing is that, let me go back one step so that we can, and then understand the implications. We know that cancer is caused by disturbances in the normal functioning of gene expression within a given cell. And it's not just one gene that has to go wrong, but multiple genes functions have to go wrong that can lead to the aberrant growth, uncontrolled growth of cells that leads to cancer. Now, there are historically, the way in which gene function was perceived to go wrong was through direct damage to the DNA where the genes reside, and that causes them to misfunction. However, in more recent times, we've begun to appreciate another mechanism through which gene function can be disrupted that can also equally contribute to cancer formation, and that is through what we call epigenetic mechanisms. And this makes reference to the title of the paper you just read, Zen, in the reprogramming of the epigenome is what was observed in this experiment. What is epigenetics and the epigenome? Epigenetics means above genetics, and it's basically molecular structures that control the genetics. So, in other words, control gene functions. And if the epigenetics is disturbed, we can have many gene functions can be disrupted without any damage to the DNA. And this is a crucial thing here. So epigenetics disturbances, epigenetic changes lead to disturbances in gene function, but without any damage to the DNA. And this is what this study found here with glyphosate. The glyphosate they found, and we'll come on to why this is so remarkable in just a short while, but the glyphosate basically altered the epigenetic profile of the human breast cells. And in so doing, it primed them. It kind of made them more readily, as it were, potentially more available or potentially more readily available to then transitioning to a full cancerous condition.
Would you call that like pre-cancerous?
Glyphosate primes Glyphosate primes mammary cells for tumorigenesis. So this is what these authors found. They found this remarkable epigenetic change in the mammary cells.
Okay.
And then that primed the cells when they were hit with a second gene, with a second carcinogenic insult. They then transitioned to a full cancerous condition.
And tell us a little bit more about that second carcinogenic factor.
Yes, this was an interesting choice. Of course, there are many second hits factors, second carcinogenic factors that they could have chosen. But what the authors chose in this study, and again, was another factor that would work at an epigenetic level in terms of changing gene function, but without damaging the DNA. And they explored a range of different types of molecule called microRNA. MicroRNA, as the name implies, are small RNA molecules. And there are literally hundreds of these within our cells of all organisms. And what the microRNA does is control gene function. And so, microRNAs, a balance of microRNAs is needed for correct gene function. But when you get, again, disturbances in the balance of microRNAs, that can lead to disruption of gene function. And that in itself, again, could be another factor that could lead to a cancerous condition. Not necessarily on their own again, but combined with another factor that has caused in itself disruption in gene function, the two combined can give rise to the cancer. And this is what was found in this study. They found that one particular microRNA, which as you can see in what you've highlighted here, Zen, the microRNA 182-5P, this has been associated, disruptions in the levels of this microRNA have been found to be associated with cancers in other circumstances. So in a way, it was a good choice here, because it was known that if you add this into the mix with something else, it could lead to a cancer. And that's what's happened here. So combining
this excuse me for a second. So this microRNA is very commonly found in human cells. Is that what you were saying?
Indeed, this particular, this microRNA is naturally found within our cells, as are many hundreds of other types of microRNA. So they are present in our cells, and we need them. It's not like it's suddenly magic, we shouldn't feel that it's foreign and not necessary. Actually, these microRNAs are needed to help control our normal gene function. But when they are out of balance, when you have too much of one or too little of another, then they result in imbalanced gene function that could be one of the triggers towards cancer. And so you're right that this, what they've done here is to artificially increase the levels of this naturally present microRNA in the breast cells, and that would have resulted in causing changes in gene function. And when that effect of this microRNA was combined with the alterations in gene function caused by the glyphosate, the cells were transformed, literally transformed to a full cancerous condition.
Okay.
And so this is the significance of this study for me, in that though the glyphosate on its own doesn't cause the cancer, but it primes the cells. So when another trigger comes along, and it doesn't have to be the microRNA, by the way, it could be something else. It could be another chemical agent that we may be exposed to.
Okay, so it primes the cells. So another way to say it might be that glyphosate exposures makes us more susceptible to cancer.
That would be a very good way of putting it, indeed. It makes us more susceptible because it has already change gene function through this epigenetic mechanism. And that has, as a result, has made the cells more susceptible in the face of a second carcinogenic hit, makes them more susceptible to then transitioning to a full cancerous condition.
Right. And I want to be clear, what we're talking about only from this study is breast cancer cells. We're not sure yet about all the other different types of cancer cells, but we do know non-Hodgkin's lymphoma, the studies have shown there's been an increase anywhere to 30 to 40 or 50% of incidences of non-Hodgkin's lymphoma when exposed to glyphosate from previous studies, but that's not what this study is about. This one's only about breast cancer. But what I want to make clear to our viewers that are watching this, because the title does say, actually it was the article in Sustainable Pulse, which I'll bring up on the screen here. It does say a second risk factor. And I just want to be clear for our viewers, or another risk factor says here, I want to be clear to our viewers that this other risk factor is not necessarily like something that is, like if you happen to be under a jet plane and the jet fuel comes down and it's an outward environmental second risk factor. It could be, as you just said, but the microRNA are also already in our body. So it occurs to me as a tiny bit misleading, because most people think second risk factor, it's like, oh, something else out in the environment that I'm not, may not likely be exposed to. So I don't really have to worry about this. Glyphosate on its own doesn't cause cancer, but you have to have some other random second risk factor. So I just want to be, clear that this other risk factor could be in your body already, right?
Yes, that's good points. I will explain.
Okay.
So first of all, the article you're referring to in Sustainable Pulse is actually a reiteration of what we wrote on GM Watch, by the way.
Okay.
So that we
can go back to GM Watch.
Acknowledge the original source.
Okay.
But you're right in that the second risk factor could be an internal factor, but the key thing here is something has to disturb its normal function. So in other words, in this case, the microRNA that is already present within us, its function has to be disrupted, and that can come from the external environment.
Oh, I see. Okay, so the microRNA in your body also has to be disrupted, and that could be from all kinds of environmental toxins. Could the microRNA also be damaged from glyphosate?
It may have. Again, there's no evidence in this study that glyphosate also alters the functioning of microRNAs. No.
Okay, they didn't test that or didn't show that.
But it may be an interesting thing to explore further.
Okay.
But no, we can't say that. But something else has to come along to disrupt the normal functioning of the microRNAs, and then added to, in this case, the glyphosate, can trigger the cancer. And I think coming back to the glyphosate, I think what's worrying for me, particularly based on the evidence in this study, is the incredibly low concentration of glyphosate that resulted in these observations of priming the mammary, the breast cells, to being susceptible to cancer.
Yeah, and so let's talk about those levels.
Very low, and that I found surprising. I have to admit to you, when as I say, I was, as mentioned in the GM Watch article, I was one of the reviewers of this article, and when I read it, it struck me that this very low concentration of glyphosate that triggered this outcome was really what makes it really striking and very worrying indeed, because this is within the realms of what you and I may have in our bodies.
Yes, and so these levels, you're talking about parts per trillion. To clarify, parts per trillion is 1 drop in 20 Olympic-sized swimming pools. And we are consuming parts per billion, or in some cases, parts per million of glyphosate in our food, correct?
Yeah, and then when you, obviously, some of that, not all of that, but even when we can take into account that only some of the glyphosate that we ingest gets into our body, but But since we are consuming at least in the United States, we're a little bit cleaner here in Europe, but in the United States, based on the surveys, we estimate that a typical a US citizen is going to be consuming micrograms of glyphosate per kilogram body weight per day. Micrograms per kilogram body weight per day. And therefore, that could quite easily, internal concentration of the glyphosate could quite easily therefore be in the range of the concentration that was tested in this study, resulting in this adverse outcome. And that's why we need to sit up and take note of the findings of this study and explore it further, because if glyphosate is able to bring about these epigenetic mediated changes in gene function at such low concentrations, we should be worried.
Yes, yeah, and it's not just an older type of cancer. What it is, it's Sophia Lelevre that says that what was particularly alarming about the tumor growth is that it wasn't the usual type of breast cancer we see in older women. It was the more aggressive form found in younger women, known as luminal B cancer. Yes. And that's a very fast growing cancer that it is clearly life-threatening and can be fatal. And this is, you know, this is the type of cancer you're hearing about in 20-year-old women. This is not okay.
No, far from it, indeed.
Yeah.
And so again, that just adds another dimension to the concern that this study raises, which is why we really need to explore further. As you said, at the moment, this has only been observed in mammary, in the breast cells. We don't know whether similar outcomes occur in other cell types of the body, but it's crying out. That is crying out to be explored, and we hope in my laboratory over the coming months to begin studies that investigate whether this type of glyphosate-induced epigenetic change in gene function takes place in other cell types. One study I could mention, we published a study a few years ago now where we treated human liver cells to glyphosate. And when in the light of this study, we went back and looked at that old set of data, we did not see the increase in the expression of the gene that led to the epigenetic changes. This is this TET3. If you go back to the title of that paper, it says the way that the glyphosate is causing the epigenetic change is through increasing the function of this, increasing the function of a gene called TET3. Okay. And so as far as we can tell so far, at least in liver, the glyphosate is not increasing TET3, but that's just one. It's possible that there are other cell types, tissue types in our body where the glyphosate may increase this TET3 and result in the epigenetic changes, the epigenome changes that are reported in this study. And we hope to explore this possibility over the coming months and beyond.
Right, yeah, it looks like there's a lot more that could be studied. And one question that came up in looking at the study was, it appears that this study tested only glyphosate technical, which is glyphosate alone. And is that correct? And not the final formulation of glyphosate-based herbicides? That's correct.
They only tested glyphosate alone. There are a number of, this study is kind of contained as far as it goes, but there are a number of gaps as well in the study, in the analysis. And one of them is the fact they didn't test a Roundup formulation at the same glyphosate concentration. I think that would have been very interesting to do so. But so far, what we see is just from glyphosate and not the formulation. And it's a very valid point to raise then, because as we know, the formulations are far more toxic than just the glyphosate alone. So it's possible that having the adjuvants from the formulation present along with the glyphosate, we may have seen an even more pronounced effect at the epigenetic level of gene function than what we're seeing with just glyphosate alone. It may have not been, it's a speculation. It's possible that it's just the glyphosate that causes this outcome, which would be bad enough, of course. but it would be very interesting in future to test both the formulation alongside the glyphosate.
Yeah, well, it is important though that it is just glyphosate, because from what we've seen in food testing, what has shown up or what we've been able to track is only glyphosate in the food. The food glyphosate gets into the plant, it gets into the cells, and we consume it. We're not quite as sure about arsenic and the heavy metals and the other petrochemicals that are in the final formulation, about whether or not they're getting into our food as well. We just don't have the studies to show that those levels of, you know, those other contaminants in the food. So this information is very useful as far as looking at the glyphosate levels in our food supply and water supply, and the potential impact, right?
Very, very true. Absolutely right. It's worrying enough that just glyphosate alone has given that, because that's so far we haven't got reliable methods available to identify and measure the formulation additives, the adjuvants in the food supply. We're working on it. I'm glad to say that my lab is working on developing the assays to look for the adjuvants in foods, and we hope over the coming months that we will have those assays in place, and we will start to explore the presence of the adjuvants along with the glyphosate in foodstuffs. And when we have that information, everyone will hear about it, obviously, and we can take steps forward beyond that.
Great. Well, I have one more question before we let you go. I know you're very busy, but there is a section in here that talks about methylation. And I know we can't be hypothetical about things, but I just hoping you could shed some light on what this means for us a little bit, because a lot of moms with children with autism have children with issues with methylation. And I don't want to extract, like draw conclusions or anything, but can you explain this a little bit? It says normally when the DNA of a gene is tagged by methylation, it is not expressed. When the methylation tags are removed, the gene can be reactivated. And such changes in gene function caused by alterations in the profile of DNA methylation tags are known to be a contributing factor to cancer formation. So when glyphosate was induced, and when glyphosate induced a several-fold increase in the protein called TET3, which is known to remove methylation tags, then we have this issue which could lead to cancer on its own, it sounds like, due to this issue with the problem around methylation. Can you talk a little bit about that, and perhaps how that might be connected to the issues with autism, or maybe just explain this? Can you explain that?
Yes, indeed. This is, again, a very fascinating area. We now know that epigenetics, as I mentioned earlier, is what controls the global patterns of gene function. And we know that chronic diseases have aberrant patterns of gene function at their basis, not just cancer, but just about any other chronic disease, physical or behavioral as well. And one of the epigenetic mechanisms of gene control is through methylation tags on the DNA. And if methylation tags are present in particular parts of a gene, and particularly a region known as the promoter of the gene, if the DNA of the promoter of a gene carries methylation tags, then that gene is switched off. And that's a natural mechanism of gene control. If the promoter of a gene does not have methylation tags, that gene is switched on, or at least it's open and available to be switched on. So as you can see from this, if we get epigenetic pattern disturbances in the DNA methylation tag profile, in other words, if we get tags removed from places where they should remain in place, or have methylation tags added where they should not be added, gene functions can go up and down in an unbalanced manner. And if the gene expression profile is not balanced and appropriate, the biochemistry of our bodies is disrupted, and that could lead to a whole plethora of different diseases, both physical and behavioral in nature. The key thing is that's been discovered in recent times is that the epigenome, or the epigenetic level of gene control, is responsive to environment, lifestyle, and environmental inputs. Not just, in other words, not just internal cues, internal signals within our bodies, but external inputs, what we eat, what we breathe, what chemical pollutants we get exposed to, behavior, our psycho-emotional state even can affect the epigenetic level of gene function. And if that results in a disruption, then it can lead to a chronic disease, whether it be autism spectrum disorder, cardiovascular disease, or cancer. All these are now known to have an epigenetic disruption component at their basis. It sounds very bleak. However, to finish on a positive note, the important thing about epigenetics, our epigenetics, it's dynamic. It's changing all the time. So if through some past bad habit or whatever, or past exposure, our epigenetics got put out of tilt and we ended up with a disease, we shouldn't feel we're stuck with it, because with appropriate lifestyle interventions, especially holistic lifestyle interventions, we can reset the epigenetics of our bodies and get back towards, at least towards, if not a complete recovery of our health. And that is, so this is really the powerful message of epigenetic basis to chronic disease, which is why what you're doing is so really wonderful to educate people in the United States, because by changing your diet, by changing your lifestyle, you know, avoiding the chemical pollutants, the pesticides in our food and other pollutants, and following a positive life, and supplementing, make sure our microbiome affects our epigenetics. So taking care of our gut, taking care of our gut biology, and all of these things, all these positive things, they have an effect. Slowly but surely, we will reset our epigenetics and move back in that direction of health and get rid of the illness.
Oh, we appreciate that so much. And we do appreciate the science that's coming out of the UK. I believe Sheepdog Trust and many different organizations there have been funding studies on the benefits of organic. And they've shown that the people who went organic, they dramatically decreased the amount of pesticides in their bodies within a matter of a couple of weeks. So we absolutely can make those changes. Monica Kruger from Germany, that we both know, has showed that the animals that had the higher levels of sickness had the higher levels of pesticides in their bodies, and lower levels. So we know that this is all connected, and the good news is, as you said, yes, thank you, we can do something about it. We can switch to organic. And for people who say that they can't afford it, I would say, let's play the farmers instead of the hospitals, and just, get your healthy organic food, and you will see your medical bills drop. Absolutely. My family spends anywhere from $15,000 to $100,000 less a year now, because we don't have children with, allergic reactions and autism symptoms. We've been able to recover our children from that. So we do not spend money on the doctors that way. And that can be the same for you too. Now, we can't promise that somebody can recover from breast cancer, but we can share this information to help prevent it from happening in the first place. And that would the biggest benefit of eating organic is that you're not allowing those toxins to get into your body and disrupt all of that, everything that you just described, all of the epigenetics that we do need to take care of. So thank you. Is there anything else you want to add before we go, Michael?
No, really, Zen. I think this is a wonderful way to finish. Great. But just only to say, yeah, always prevention is better than cure. And this knowledge, what we're doing with this knowledge, we're empowering people to take control of their lives. They don't have to be at the mercy of what the polluters are subjecting us to. We clean up our diets, we clean up our life, clean up our homes, make sure we don't buy these toxins, cleaners or whatever into our homes. And we will, as you say, we will see the difference. And the wonderful narratives of families like yours that have experienced this transition from a state of illness in their children, especially to health, is really what is inspiring for me, and I hope inspiring for others to follow your example.
Yes, and you can find out more of, I don't have the book in front of me, but you can see it behind me, my book, Unstoppable. We do quote Michael in that book. And if they want to support your work, Michael, how would they do that? Are you privately funded, or do you get funding from universities? How does your work get funded? Just so.
This is a good question. It's almost impossible to get money from governments to do the kind of work that we do. So believe it or not, I'm very beholden on actually a major philanthropist. At the moment, I get most of my money from a philanthropist in the USA, and God bless him. You know, without him, I'd be doing nothing. So, so yes, we would welcome donations to expand our work all the time. I mean, there's always 10 things to do, and we're fortunate to do one or two.
Yeah, one or two studies. So would they go to the King's College of London to look you up and to contact you, or how would they do that?
Yes, if there are people interested to support work like along the lines of what we've just been discussing, looking for these, say, glyphosate epigenetic effects, we're ready to go with those. If resources were to come our way, they should just contact me directly, and we can discuss ways that their support can be channeled to me at King's College.
Okay, and how would they contact you?
Oh, via email.
The email. Okay. So, and that is Michael Antoniou. Let me see. Right.
They could
Antoniou.
contact me at my work email address or
Okay,
my.
which one is that?
That's Michael.Antoniou. This is all lowercase.
Okay.
So it's my name, Michael.Antoniou at kcl.ac.uk.
KCL dot what?
.ac.uk.
Okay, great. Thank you so much. We'll put that at the bottom of the at the video as well. Thank you very much for your time. It's a pleasure to see you. We appreciate your review of this very, very important work. This really is an astounding study, don't you think? And really crucial to the future health of women and people around the world. Thank you so much, Dr. Michael Antoniou. We hope to see you again soon. Take care.
Indeed. All the best
Bye.
to everybody. Yeah.
Yes.